Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few hands on, we have done the differential expression analysis for a RNA-seq data set, right. We have used DSEC2 and then subsequently we used a data set with the spike-ins in there, ERCC spike-ins, which we used as a control for our normalization. We used RUPG method using the RUPSEC package. Now, we have the results. So, what we want to do is we want to look at the results and maybe visualize some of this. We have used MA plot already, but maybe we can try the volcano plot, how to create this volcano plot and maybe a part of the data as heat map. Okay. So, we have talked about this in the theory class. So, I would like to actually show you how we can generate these plots. And then followed by that, we will talk about the functional enrichment analysis very briefly using a package in R again. That will give you some of these enrichment results. Okay. So, that is the agenda for this class. We will try to visualize some of these results that we get and then do the functional enrichment analysis using an R package. And here are the steps, right. So, as you have seen, we have now completed up to the differential gene expression analysis. We have also got the results and we have seen the results from two types of differential expression analysis, right. So, one without spike-in, the other with spike-in, and we have done the plot, the MA plot, right, for this analysis. What we are going to do today is we are looking at the volcano plot, so and the heat map, and then functional enrichment analysis. The tools that we are going to use for heat map, we will use this gplots package. There is a heat map 2.2 function that we will use for our analysis. And for functional enrichment analysis, we can use different types of tools. We have mentioned different tools in the theory class. What we will use in this class is the R package gprofiler 2. This is a package that utilizes the hypergeometric test. Okay. So, it is a hypergeometric test based enrichment and we have talked about this in the theory class, the hypergeometric test, how does it work. And here is the link to the package and the references if you are interested in the implementations and the actual package, okay, how it is done. So, you will see many of these tools are available as servers, web servers. So, you can simply list your genes there, put the list in the server and you can select perhaps the organism name, right, and then you can get the results very easily. But sometimes you probably have to do this for a large number of samples repeatedly, and in that case that becomes very tedious, right. So, in those cases you probably would prefer a command line based tool that you can use, right. So, a package perhaps like gprofiler where you can automatically do this analysis for a large number of sets. Okay. So, this is the tool we will use and we will go step by step. Okay. So, the first thing we will do is the volcano plot. We have talked about this in theory, and let us now go to R and we will actually do the volcano plot first, right, and then followed by heat map, okay, and then we will move on to the functional enrichment analysis. Okay. So, for the results part, right, I can simply show you the results, right, raised to, right, this is what we got after the last analysis. Remember, we have this RUPG method, then the DSEC2 we have run, and this is the final results that we got. Okay. So, what we want to do using these results, we want to generate this volcano plot, right. So, you remember volcano plot, right. So, what you have in volcano plot, you have this p value, right, log minus log 10 p value in the y axis and in the x axis is the log fold change. Okay. So, this kind of gives you, right, the significance of the results or significance of this log fold change that are observed for genes. Okay. So, what we have to do first, right, we have to first load the data. Let us see what we get. So, as I have told you, right, in R we can access these results using this dollar sign, right, what are the results that are available. So, we have this raised to dollar log to fold change. I can simply right, paste it here, okay, but that will give you all these values for all genes. Okay. I can also just check, right. So, for genes you get these values, and then you can also look at the p value, right. If you have p values there, we can also, yeah. So, we have p value and we have sorry, just giving p adjusted. Okay. So, maybe we can sorry, yeah, Yeah. So, here it is, right. So, you get p adjusted, or if you just want the p value, not the adjusted one, you can try this. Okay, so you can try anything, it does not really matter. So, what we will do is to generate this Volcano plot, we will first export this, right, export this data into some variables or assign some variables for this, maybe not x, right. So, or maybe x value, because x we have used before already. So, x value we can simply assign this data log to fold change, okay, and y value is, remember, minus log 10, and then we have p adjusted, we can say that is okay. All right. So, now we have calculated this and we have assigned the two x val and y val. What we have to do now, we have to plot these points, right, and generate the graph. Okay. So, the command for doing that is very simple, right. We can say x val, y val, and this is something what we get. Okay. Now, the problem, as you can see, right, most of the points are clustered around here, there are some outliers, and perhaps we can shift this axis a little bit, right. We can change the range of y axis, because that would be better, right. So, we will do this, and we can make a nicer plot, right, from this really basic one. Okay. So, let us try that. Okay. So, what we will do is we will change the limits of y axis is from 0 to 10, let us say 10. Okay. Above 10 we have very few points. So, maybe these are not really critical for these visualizations. Okay. And we can see, right, this. The other option would be to make it logarithmic. Okay. That will change the shape of this curve. And similarly, for this x axis, right, as you can see, most of these values are contained within -3 and +3, right. So, we can change the range again, and this kind of gives you a very nice shape, right. And this is expected. Why? Because the genes that show very low log fold change, right, they are unlikely to be significant in terms of statistical value. So, maybe it is better if we just also label this x axis and y axis, because then that will make things clear. What is this x we are looking at, right. So, log 2 fold change we can simply write, and y lab equals to minus log 10 p dot adjusted. So, because we have taken the adjusted p value, right, and we can also give a main, right, which is, let us say, Volcano plot. All right. We can change the font size. We have talked about this in the last class. So, simply saying there. All right, and this looks a bit nicer, right. We can also put the thresholds for this p adjusted, right, because if you set, let us say, FDR at 0.1. So, we are looking at minus log 10 p adjusted to be 1, and maybe we want to put this cutoff and show like which genes are above this cutoff, right. Because when you transform this p value by minus log 10, right. So, higher value of minus log 10 p adjusted means more significant results, right. So, we can set the cutoff now. So, this will be using this command, right. So, every line, because this is a horizontal line, right, we are setting a cutoff for minus log 10 value. So, this is a horizontal line. So, you can use h equals to 1. Okay. We want this in different color perhaps, and also maybe dotted line. Okay. So, this is something we can achieve by just saying color equals to blue. Okay. And here is the cutoff that you get. Okay. Now, it might look a bit nicer, right, if we could try, right, to make these points with different colors. Okay. So, for example, some of the points may be the ones that are not significant, maybe they are gray. So, the points that are significant, they are in other color, let us say red or something. Okay. So, to do that, first we can try is something like this, right, which is h equals 16, right. So, this will change this open circles to filled circles. We can say color equals to RGB, right. So, we have talked about this before. we are setting this RGB scale, right, and this looks a bit nicer than before. Okay. And it would be even nicer if we can get different colors for them, right, and this is something what we will do now. Okay. So, for doing that, what I will do, I will take a very simple kind of step. Okay. We can have, let us say, first I will color these points as white. Okay. Why I am doing that, you will realize in a moment. Okay. And then on top of this, okay, in R you can actually, on top of a plot you can add points, okay, and you can keep on adding points in different colors. Okay. So, that would be great. So, first let us say, let us add this line. Okay, I will add this line now, this is the cutoff. So, for adding points, the command is this. Okay. So, x which x values, sorry, y values are less than equals to 1. Okay. So, I am plotting only those points now that show this, for which the y values are less than equals to 1. Okay. This will give me the points that are below this line. Okay. And similarly, for y value we will choose only the ones that are below 1. Okay. PCH equals to 16, right. Now, we want the colors, we do not have to add any axis labels etc., because these are already there, and then have this color equals to RGB. Okay. The last one is for transparency. Okay. So, you see we have added these points that are not significant, okay, and these are in gray color, they appear black because there are so many points in there. What we want to do is, now we want to add these points that are on top, but on a different color, maybe red or something. Okay. So, let us go ahead. Okay. So, we will just simply change the condition, we will use the same command. So, points, right, we are adding points to the same plot, we are not creating a new plot. Okay. If you create a new plot, these points will be gone, right, and you have to also add the axis etc. The only thing we need to change now is the color. Okay. So, maybe we can add this color, right, in red, these points, and let us see how it looks like. Okay. Okay. So, I have just changed the colors here, you can see. So, this is for red. So, I have said, okay, red intensity 0.5 and then also transparency 0.5. Okay. And this is what you get, right. You have seen this seen this in the theory class, right. So, I have shown you some of these figures and it looks something like this. Okay. So, you have these points in different colors and you can then easily identify which are the genes that show difference in expression, significant difference in expression in these two different samples. Okay. Now, you might, in some cases when you have a lot of genes, in some cases you might want to color them based on their log fold change as well, right. So, maybe you want to set up a cutoff of +1 and -1 and you can change this color again. Okay. So, this again you can do is in the same way. I am not going to do that, but you can do this in the same way that we have done. Okay. Only thing you need are more conditions here, right. So, instead of this just saying which y value greater than 1, you have to add also AND where log fold change or the x value is greater than 1 or minus than 1, right. So, that kind of conditions you have to set and you can get different colors. Okay. And you can of course save this, right. We have talked about how you can save this in R in PDF format. We prefer the PDF format because of its resolution. So, it will maintain the resolution, otherwise if you export in JPEG or BMP you would lose resolution. So, you can simply save this using this dev.copy to PDF, right. So, this output will be copied and then switch off. Okay. So, this part is done, right. The next thing you can do is the heat maps. Okay. So, for heat map we can use this package called gplots. Okay. I have installed this again and it is very easy to again install. You can simply, if you say bioconductor package, right, you can simply go to bioconductor page and use this BioSim manager. Okay. And in this package we will use this heatmap.to function. Okay. This is a function that we will use. It has a lot of option and the usage is given, right. it will have a lot of options, but do not worry about this. It will start with a very simple thing from our data set. Okay. So, again I have prepared these commands, right, which we will use for generating this heat map, okay, because I want to show this in a very short period of time, right. So, we will load this package library gplots. Sorry, there is a spelling mistake. Yeah. So, here is a gplots package we have loaded, and now we want to use this heat map function. But for heat map function, right, so first thing you require is this x to be formatted properly. Okay. So, what is this x? X is this matrix of these values that we have to plot. Okay. So, in our data what I have done, right. So, for heat map, as you have seen, we want to look at different samples. So, what I have done, I have taken this count data, okay, and I have created a file which you can use for doing this heat map analysis. Okay. So, I have created this heat map example data, as you can see, with taking only a few genes, okay, and their expression values. This could be raw count or normalized count. The process of heat map generation is the same, right. So, we will load this heat map example data in R, and then we will use this heat map dot to function to actually do the heat map, to generate the heat map. Okay. So, let us load this quickly. Okay. So, here it is. Okay. As you notice, there is no header. So, I have not given the header. So, the default option is false, and if there is no header, R will add its own header, right, V1, V2, etc. Now, what I need to do is we need to convert this to matrix, right, and we also need to get rid of this first row or the gene names, right, because heat map to function expects only a numerical matrix, only numeric data. Okay. So, to do that, we will do this, we will do this conversion. So, as dot matrix, right. So, we are getting this data as matrix dt minus 1. So, we are removing the first column. So, minus 1 means we are removing this first column here, because this is the gene name. We do not need this, or heat map 2 cannot use this information, okay, and we have generated this new data. Okay. So, and if you can check this, you will see only these values are now remaining. Okay. And since this is a matrix, we can now use it heat map function, okay, and we can simply say dot 2 dot x. Oh, so it is still giving, oh sorry, not x, right, yeah, dt 2. Okay. So, here it is, we got the heat map, okay, and in the heat map what you have along these rows, you have the genes, and along the columns you have the samples, okay, and you have some sort of clustering that is done here, okay, both on the samples as well as on the genes. If you do not want these clusters, right, you do not want this clustering, hierarchical clustering, you can switch them off, and you have the option to switch it off along one axis. You can say do not cluster along these gene names, or do not cluster along the sample names. Okay. So, sometimes we do not prefer to cluster along the sample names, because we do not want to change the order of the samples. Okay. So, that is something you can decide, you have that option. Okay. So, what I will do, I will make it a bit nicer. One of the things you will probably see, there is some lines that are going through this heat map. So, first let us remove those, okay, and you can see the scale here on top, right. So, red means negative and or is close to 0, yellow means positive numbers, like large positive number. Now, in heat map, when you are generating heat map, one thing you would have to be very careful about is that you are working with genes that show very high degree of difference in their expression pattern, and if you are using those genes for clustering, right, the ones with very high expression will dominate the clustering process. Okay. So, to remove that effect or to minimize that effect, what we do is we do something called scaling. Okay. So, scaling will bring all the gene expression across samples to the same level. Okay. So, all these genes will get similar weight in the clustering process. Okay. So, by clustering, we just want to see some of these expression of genes that are very similar to each other across these samples. Okay. So, what I will do is I will use this scale function, okay, and you can see now this looks much nicer. Okay. we have this transformed because we have scaled, and you can see this Z score because this is after the scaling, the new score that has been calculated, and this is for the different genes. Okay. So, we can now compare these expression patterns across these different samples. One of the last things I want to do is I want to switch off the clustering for the samples, because as I said we do not want this, right. So, we can actually switch off this using this cluster, right. So, dendrogram we do not want, I think, and then we can also switch off the clustering somewhere here we have to look at again. So, there would be an option to actually switch off this clustering, so that you do not get this reordering of the sample. Okay. So, this is again maybe I am not able to find this right now, but you have this option where you can actually do this for your sample. All right. So, let us now move on to the next part, which is the functional enrichment analysis. Okay. So, we have now seen how we can generate this heat map, right, how we can generate this volcano plot, and of course, there are other options, other visualizations that you can make and you can explore, okay, but we will not be able to cover all those in the limited time that we have. Okay. So, the next part is the functional enrichment analysis. Very briefly I am to show you how we can do that using a package called gprofiler 2. Okay. So, I will switch off or remove this window, right, and clear the screen. Okay. So, let us see the package, the gprofiler package here. Again, this is a package that you can download very easily or install using this R console, right, and you have the manuals, etc. All the details are here, and here is the web page. You can see, right, all the information is available and in details here. Okay. So, the main function that we use is this GOST. Okay. So, this is the enrichment, and all the instructions are given. You can see the installation instruction, the library you have to load, and the function that we will have to use is this one. Okay. So, this is the result that we get. The function that we use is GOST. The query list, this will be the list of genes you can provide. Okay. You can simply mention the organism name, the short name, right. So, this is H. sapiens, this is the human data. In case of working with where we are working with Saccharomyces cerevisiae data, we can simply say S. cerevisiae. Okay. So, these naming conventions are also given in another page, we will see in a moment. Okay. Then you have to you also have these options, different attributes that you can use. For example, you can use ordered query. So, this is false, means whether you have sorted this list according to, let us say, log4 change or something, right. If that is the case, you can say this true. Whether it is multi query, right, you can also have multiple lists that you can give, we will see down there, right. If you have multiple lists, you can also give, you can also exclude some of these annotations, right, in the geo which are in silico annotations. Okay. So, maybe they are not very reliable, so you might not want to use those annotations. You also have this measure under representation, which means whether you want to measure over representation and under representation, that you can specify using this, right. You can then choose the threshold p value, right, that will use the correction method. This is a multiple hypothesis testing correction method. Here the default is a method for this package that is designed by this package. You can also change it to FTR, and then other options you can see, right. Again, these are also explained in this page. Okay. So, what we will do is we will take a list of genes, right, and we will try to do this run and see what we get. Okay. Again, this is all explained in very detail here, and for multiple queries also you have these options. You can somewhere down there it is also written. Again, you can see this query of different query lists, right. So, different lists that you can also have this, and you can say multi query equals to true. Okay. Now, coming to the organism name, right. So, if you are working with a specific organism, right. So, let us say you are working with Saccharomyces cerevisiae that we are working with, right. We can simply search, and this is the data that is already present, and you have to use this S. cerevisiae. Okay. So, this list is also part of the gprofiler package, the web page, right, and you can find this list and then for different organisms, and these are given here. Okay, so what we will do is we will run this using this GOST command from a with a list of genes, right, random list of genes that we find. Okay. What in reality what we will do once we have identified these differentially expressed genes, let us say we find let us say 100 or 200 genes that are differentially expressed and we have filtered them in all possible ways, right, then we want to do this functional enrichment analysis. Okay. And so on, what we will do, we will create this list of these differential expressions, right, and then give it to this program, okay, and which will then identify the function classes that are enriched in this process. Okay, so what we will do then is let us go to the R console, okay, and I have already installed this gprofiler 2, right. Of course, it is giving me an error, that is giving me a warning, not an error. So, that is okay, it is just saying perhaps we need to update this R version because this is not the latest version, the one that I am running, but it is okay, it will run. And then what we will do is we will have a look at the list. Okay. So, the list of genes that we got, and we can perhaps choose at least some of these genes and we can create a list and then give that list for this analysis. Okay. So, I am choosing this gene in random, we do not have time to find out which genes we will choose because sometimes that requires a lot of careful considerations, right, and maybe identifying those genes that you think are important, right, after those filterings, log fold change, etc. So on. So, once you have created this list, you can once you have the data, you can create this list very easily, right, and simply load that list in R, right, rather than writing or creating this like a list like this. Okay. You can simply load it and then you can give it or give it that list as an input to this program. Okay. So, what I will do, I am just copying some of these genes here, gene names, as you can see, and perhaps we can send this to this program. Okay. So, here we are. Okay, I think this should be okay, right. We have given 5 genes and let us now run the analysis. Okay. So, how do we run this analysis? Okay. So, again we have I have this code. So, just to refer, right. So, the results that we will get, right, the enriched results, sorry. Okay. So, equal to GOST, right, then query equals to list, right. In case you are loading this, you just simply give that variable name where you have loaded your data, the list, and simply specify here, and then we have to say organism name, right. So, this is required because the program will then search, right, these gene names and then associate them with specific functional classes, right. So, or maybe click classes, right. So, here it is geo classes, right. So, this information is required because then the program can find these genes and associate them with the function classes. Okay. And the second part, it will also look at the global list of genes and their functional associations, right. So, if you remember the hypergeometric test, you remember, right, we have a global list and the a sample. Okay. So, the sample in our case is the list of differentially expressed genes. Okay. So, the rest of the things we really do not need to mention, right. So, unless you want to change them, they will be taken as default values. Okay. And only thing we can perhaps do is we can also look at this correction method, right. So, maybe we can change that correction method to FTR. Again the program mentions, right, what are the viable options. You can also give Bonferroni correction. Okay. And we have got this now. Okay. What we want is perhaps okay. So, enriched results, names of enriched results. So, as you can see, we have found this the variable names that are present, right. So, Enrichment result. So, you have this query, this is the least p value, right, and term ID, term name which are given. Okay. So, what I will do is then I will have a look at the first few lines, let us say first 10, okay, and you can see, right, this part of query 1, okay, is significant, whether it is significant or not, there is some p value, right, and then you have the terms names, right. So, you have the geo ID, right. So, you have this term ID, this is the geo ID, right, the source is GeoBP, the biological process, we have again talked about this sub-ontologies. Then you have the term name, right, these are the functions that are associated with these genes of this query. Okay. So, we can now expand this, right. Of course, you can take bigger lists and then we can identify the functional classes also again according to the biological process and so on. There are other tools as well you can explore, but the idea is very similar. You can have these hypergeometric test based tools, right, or you can go with the GSEA like over representation analysis, right. So, that is it for the hands on of RNA sequencing data set. So, we have covered a lot actually. We have started with the data mapping, right, where we actually mapped using HiSet2, and we have done the raw counting, right, raw count data we got after using HT-sec count, and in this set of hands on we actually process the data through all the steps. Okay. So, from bias correction to normalization to differential expression analysis and finally to functional enrichment analysis. So, from the next class we will be talking about the genome assembly and how we can use NGS data for the assembly process and what are the algorithms that are available for assembly and which one is the preferred one. We will talk about that from the next class, and finally, we will talk about the epigenetic modifications, study of epigenetic modifications using next generation sequencing technologies. Thank you very much.